There's one other thing that geneticists use genetic mapping for. This is something that I like to call phenotype mapping. It's not just genetic mapping as in calculating genetic distances between genes. That might seem like a rather trivial exercise to you, despite the fact that I've now spent like 3 classes talking about calculating genetic distances between genes. The goal of that, again, is just to estimate whether or not genes are on the same chromosome or different chromosomes. Maybe to you that's boring. To me, that's kind of boring also. But it's important to have that skill. In this case, this is a bit more maybe relevant. I'm sure some of you can think up of reasons that this is relevant. Trying to figure out what genes control certain traits. Phenotype mapping. We want to know what genes control certain traits that we're studying. So here's an example. This is called a simple trait. The word simple here means one gene. So if I talk about a simple trait, I'm talking about a trait, a phenotype that's controlled by genetic variation of a single gene. If we're talking about a complex trait, which is the next thing we're going to talk about, that's a trait controlled by multiple genes. So what's an example, do you think, of a complex trait in humans? You've looked ahead in the slides, but it's absolutely right. Skin color, eye color, height. What would be true? Hair color. Yeah, absolutely. A lot of our phenotypes are controlled by multiple genes plus the environment. It's not just a single gene controls the trait. What would it be like? Imagine, close your eyes. Imagine what the world would be like if human height was controlled by a single gene. And there were 2 alleles. What would the human population look like? You'd be tall or you'd be short. Or maybe you'd be medium if there wasn't simple dominance. So if you're a heterozygote, maybe you're medium. Right? So there'd either be 2 or 3 heights of people. That's not what we see. We see that there are a continuous number of heights, which tells a geneticist one of 2 things. Either that trait is controlled by lots of genes, not just one, because if there was just one gene, it would be binary, or the environment also plays a role in height, which we certainly know to be true. So in this case, a simple trait. We've got one gene, the gene is called A, up here in the corner. And so there are 3 different possible genotypes. You're either homozygous big A, you're heterozygous, or you're homozygous little a. And you can see the cartooned skin color, hair color, whatever you want to call it, phenotypes. Those are the colors of the boxes. So you have 2 black alleles, then you have dark skin color. If you have 2 of the white alleles, each of those dots representing a little a. Then you have light skin, and the heterozygote has an intermediate color. So you see there's the 1 to 2 to 1 genotype and phenotype ratios you expect from a monohybrid cross. That's simple. That's why it's called simple mapping. You get a perfect correlation between the genotype and the phenotype. That's what we're looking for, by the way, in the upcoming slides. One genotype corresponds exactly to one phenotype. So you have 1 black allele of this gene, you've got an intermediate skin color. If you've got 2 of the black circles, 2 capital A's in this case, you get the darkest skin color, hair color, whatever you want to talk about. So our goal is identifying the gene. So this is a case where we know the answer. This is what you expect when that gene controls that trait. This is what you would experience if you were actually doing this sort of a study in an organism. So here I'm diagramming the phenotypes of 8 different individuals. Except I'm not telling you which of the genes controls this skin color trait or hair color or whatever. So for 2 minutes till 11:45, figure out which of those genes, B, F, D, C, or E, controls this trait. This is a trait that's where the phenotype is controlled by one of those genes. So, is it E? Well, I think it's E because the individuals that have dark phenotypes each have an allele 1 of gene E. And each of the individuals that have light phenotypes have at least one E2, the second allele. So why is that wrong?
Those two have to be homos, I guess, at each one.
If there's what if there's what if there's dominance? So yeah, what evidence do you have?
Looking at the first individual and last individual, they both have the same combination.
Yes, so that's one way that you rule out that E is associated with that genotype, gene E is associated with that trait. Because if you get the same genotype in two different phenotypes, can't be that gene. Okay, what else? What's the answer? F. Okay. So in F, what did you notice?
The two with dark skin have the F1, and then the light skin have the F2, F2.
And that's what I'm talking about when I'm talking about just looking for the perfect correlation between the genotype and the phenotype. Every time you see one phenotype, it has one genotype. So which one's dominant?
Most of them, based on what we did earlier, the F1.
Well, it's actually, it's what? It's codominant, which is not something we've spent a lot of time, if any, talking about. But the heterozygotes have an intermediate phenotype. So there is no simple dominance happening here. This is codominant. Okay. So fantastic job. That's exactly how phenotype mapping works. You look at the phenotypes, and then you figure, this is how we know, for example, that gene F controls this trait.